This is the beginning of chapter 12, which is all about signaling. So biosignaling is any time where we have a signal of some sort that causes a chemical change. So this usually is going to happen, the signal will be outside the cell, and then the eventual chemical change that leads to what we want will be inside the cell. So because of that, most of these signaling pathways are going to have at least some portion of something that is found in the plasma membrane. So we're first going to talk about some general things that are true for most signaling pathways, and then we'll get into examples. So one thing that is usually true is that there's a high affinity between the signal and its receptor, and that is simply so that you don't have to have very much signal to start the pathway. So if we're looking here at this signal that's going to bind this receptor, the KD of that binding affinity is usually around the 10 to the negative 10th molar. So that's about 100 picomolar. So remember, smaller is better. That's pretty tight. Okay, it's also very specific. So you might have this sort of similar substrate that may not bind the receptor. That's what they're showing here. Because this binding of the receptor into the signal is going to eventually lead to a response, and you don't want to have to make a ton of this signal. And you also don't want it turned on at the wrong time by a similar signal. Another thing that is very common is that amplification may occur through a cascade. And what all that really means is we have multiple steps between the initial signal and its enzyme or its a receptor or whatever, and then later what the downstream effect that we want to have. And the reason for this is it just simply makes things faster. So if we have this signal that acts on this enzyme, if this was the final thing we wanted to happen, it's limited by how many enzymes got affected by the signal. But if instead this enzyme can maybe act a few times and make three different molecules, or three of the same molecule, but 3 times, and then each of those can go act on a second enzyme, and then each one of those makes 3, and each of those acts on a second enzyme. This is sort of like how memes can spread through the internet, right? If you share an image, and 3 of your Instagram friends share that image, and then 3 of their friends share their image, right? Like pretty quickly, we get really big numbers of people who are sharing the image. It's the same thing here, versus if you tried to just, as a normal person, share an image and get 10,000 people to see it, that's not likely to happen, right? Unless you're like famous. But it's a lot harder to do that, whereas getting something to spread is easier. It's the same idea, right? So this is just called amplification, and the cascade is the fact that it goes through multiple steps. Another thing is most pathways are going to have a way to turn it off while the signal is still there. So desensitization is not the same as saying the signal is gone, we should turn this off, okay? This is saying while the signal is still binding to the receptor, if this happens long enough, there will be a different response that's going to go say, okay, calm down, like this is just maybe here forever, okay? We have all probably experienced this with our sense of smell. So if you have ever like come back to your house after a while, you're gone for a week, and you go, huh, my house smells a little different, right? The reason for that is it's always smelled like that, and you just didn't smell it anymore. Or if you've ever had to do a particularly really smelly job for hours on end, at the very beginning you're like, this smells terrible, and after maybe an hour or so, you're like, oh, this is starting to smell better, and somebody else might walk in and be like, woo, smells terrible. Again, it's because there are signaling pathways that tell your brain what you're smelling, and if they're around for long enough, you basically stop paying attention to them. And this is because of desensitization. It literally turns it off. So many of them are going to have this. The reason for this is it's just not worth the energy. If this path thing is going to be around for a really long time, you don't want to be constantly thinking about that smell or whatever it is. Smell is, I think, one of the most obvious that we can notice, but it happens to many, many signals. A little bit more complicated, and I'll try to explain this more when we get to examples of it, is that we also have modularity, which essentially means you don't have to reinvent the wheel. So we have certain things that can maybe be found in more than one pathway, and they're very similar. Maybe you have something that needs to bind a phosphorylated residue, and so instead of having a completely different thing every time that does that, there are sort of families of proteins or signaling molecules that can do this. And it kind of just allows it to just, again, be not really reinventing the wheel. But I think this is a hard one to understand without an example. So when we talk about examples of that, we'll get into it. And further, we're going to talk about this most of the time as here's this pathway and here's how it works. And it's going to seem like it's kind of all on its own. But all this stuff is happening in cells. There's a lot of stuff going on in cells. And integration of more than one signal can and does absolutely happen. This can happen in two ways. One could be you need both signals to occur for your eventual response. Another could be that one signal will turn on the response sort of. The other signal by itself will turn on the response sort of, but if you have both, you have a really high response. All right, so this might sort of, again, try to have an analogy here. If you haven't eaten in 12 hours, you're probably going to be hungry. You consider that maybe that's probably one signaling pathway. Alternatively, if you go and run 5 miles, you're also probably going to be hungry. However, if you go and run 5 miles having not eaten for 12 hours, you're going to be ravenous, right? And that's because of the integration of the two pathways. It's a little more complicated than that in terms of eating because it's a lot of crossover, but it's an analogy here. All right. There are also times when you might have one signal that turns something on and a different signal that might turn it off, so they can have kind of the reverse. But again, I just kind of want to point out here, things are related, even though we're often going to try to focus in on one so that we can understand it. It gets really complicated when you start trying to think about too many things at once, but everything is in cells and everything's possibly happening at the same time. So we are going to group human receptor pathways into 6 different main types. I want to be clear here, this is not all the possible types of receptors that could ever exist. These are the 6 most important ones that there are groups of, and this is specifically humans as well. We'll see a couple examples at the end of the chapter for things that are in bacteria or plants, and we'll see that they don't actually fit neatly into these categories either. So I just want to, don't think that there can't possibly be a pathway that doesn't fit into one of these, but this does categorize the vast majority of human ones. So we're just going to go through these kind of in order, and they're in the order of how much time we're going to spend on them, to be honest, too. So we're going to spend a lot of time on the G protein coupled receptors. And the reason for that is that there are just a ton of them. They're really important. Humans have about 1,000 different G protein coupled receptors that all bind different substrates. They're involved in our senses of taste and smell. They're involved in detecting hormones and growth factors and things like that. A lot of drugs are like the pharmaceutical drugs that you might take for various diseases or issues or whatever. A lot of these are designed to work against G protein coupled receptors, so they're really important. So we're going to talk quite a lot about them. And that's going to be the rest of this video. So broadly speaking, again, there's 1,000 or more of them. So broadly speaking, and then we'll get into a couple examples. So G protein coupled receptors, there's a receptor in the membrane, and then there's a G protein that is coupled to it. So this green guy is called a G protein. And these G proteins, the ones that are bound to GPCRs, are usually have 3 subunits, alpha, beta, and gamma. And so the way that this works is when the GPCR is not binding its substrate, it is going to be bound. So we got the GPCR, and the entire G protein is going to be bound here, and it's also bound to a GDP. So the reason it's called a G protein is because it binds and cleaves GTP to GDP. So this guy is just sitting in the membrane, ready, waiting for a signal to come along. So the signal comes along, and we're being vague here because, again, it could be any number of signals, and the signal is going to bind in the GPCR. So the GPCR has generally 7 transmembrane helices, so it's one of those type 3 integral membrane proteins where we can see how it could maybe like nestle, the substrate sort of nestle in here. And when that happens, it's going to change the configuration a little bit, which is going to cause this alpha subunit of the G protein to let go of the receptor, also let go of the GDP, and it's going to do two things. It is going to pick up a GTP instead, which activates it, and then it's going to move elsewhere in the membrane, usually not very far, but to something called an effector enzyme. All right, and this effector enzyme is going to do some sort of chemical reaction, catalyze some sort of chemical reaction that leads to a signal. Again, these are various different signals depending on the pathway. And after a while, this G protein is going to cleave the GTP back to GDP, at which point it becomes inactivated, and it's then going to move back to its partners over here, the GPCR and the beta and gamma subunits. If the signal is still there, this could happen again. It could get, reverse, go back around. But if it's gone, it'll probably just stay there and wait for the next signal. So then the signal is going to cause further downstream amplification, and we're going to have usually many steps after this. But this is the first step. The bind G protein moves over, affects the effector enzyme. So the effector enzyme is only going to work when it's got an active G protein alpha subunit here. So the one we're going to talk about, for the most part, is going to be the beta-adrenergic receptor. And adrenergic receptors are also actually a class that bind epinephrine, which is also known as adrenaline, same compound. It's this guy right here. And so that, of course, is the signal that our bodies create when we are suddenly scared. So it causes us to have a fight or flight response. So this, in the past, might have been, a predator jumps out at you, a tiger jumps out and you have to either run away or figure out how to fight the tiger. These days, a lot of us have probably felt it if, you're driving your car and suddenly somebody swerves in front of you. You kind of get that, like, your heart starts beating really fast, your breathing starts fast, you're very awake. All of that is the response of the epinephrine receptor pathway. So there are multiple adrenergic receptors, and they're classified by their ability to bind to both epinephrine as well as an agonist and agonists and antagonists. So just briefly, all that means, agonists are molecules that are not the native compound that should bind that, but can bind and activate the pathway. So this guy right here can bind and activate the epinephrine pathway, even though it's not the one that normally does. And then this propranolol, which is an antagonist, it can bind, but it will not activate the pathway. So this is kind of like, it can act sort of like a competitive inhibitor, because if it's bound, then maybe the epinephrine cannot, right? Especially when it often has a much smaller KD here. So we're not going to worry too much about what these guys do and why they make it the beta-adrenergic receptor, but that is the reason it's called the beta-adrenergic receptor that we're going to talk about. All right, so the specific mechanism here, again, we've got our beta-adrenergic receptor, which is a GPCR. It's bound to its G protein. Sometimes these guys have special names. This one does not have a special name, but they're all just G proteins. So when epinephrine comes along and binds, the alpha moves over here, picks up a GTP, and activates, in this case, the effector enzyme is called adenylyl cyclase. So it's a hard word to say, but all this does is it cyclizes ATP into cAMP. So we saw these back when we talked about DNA. And so what's happening here is, here's our ATP, and we're going to have the 3'OH. It's going to come up here and attack the phosphate and kick off pyrophosphate. So pyrophosphate is just what we call it when we have two phosphates stuck to each other but nothing else. So inorganic phosphate, but two of them. So PPI is the term we usually use for that. So it's going to kick that off, and that leaves it as this adenosine 3'5' cyclic monophosphate, which is called cAMP for short. It's the most important cyclic one, so it got the name. All right, so cAMP is then going to be what's called a second messenger. So essentially you would consider this the first messenger, and then the cAMP is the second messenger. And one reason that we need this, aside from the fact that it's always just good to have multiple steps because of the amplification, is because epinephrine can't pass through the membrane. So if it's out here in the outside of a cell, it can't possibly do things on the inside of a cell. So what we do, this is going to transfer the message from the outside into the inside, which is where CAMP is. So CAMP then is going to continue the pathway. This is all one figure in your book, but I broke it up so you could see it. So CAMP is going to activate an enzyme called pKa, and this just stands for protein kinase A. And we know that kinases are going to put phosphates onto other proteins. Protein kinase A was one of the first ones discovered, so they just named it that. It's a pretty broad-ranging one. It puts phosphates onto many different proteins. And the way that it activates, the way the CAMP activates this is a cartoon of protein kinase A. So it's got, actually this is not actually part of protein kinase A. This is something that's going to hold it together and with other things. We'll see this in just a second too. So we have this, it's a dimer. And so we have this portion right here, this R domain, and then we have a C domain, which is the catalytic subunit. And the catalytic subunit is going to, this is this sort of cleft in here. You can kind of see that the red thing is sticking into. That is where the active site is. So this is the way it's structured before the CAMP comes around. And you can see the active site is hidden. It can't do anything. CAMP binds over here to the R subunit, the regulatory subunit, which causes it to let go of the catalytic subunit. And now these little things mean it's active. All right. So this is actually pretty common with kinases, because what kinases do is they bind other proteins. And so it's actually really easy to get them to also bind to another portion of their own protein and auto-inhibit it, unless you have some sort of activating molecule like CAMP around. So we've got, now we're active, and now this pKa can go and do further steps in the pathway. So pKa, like I said, it's a kinase. That's what the K stands for. Generally a pretty good bet that if K is somewhere in the abbreviation, that probably stands for kinase. I'm sure there are situations where that's not true, but it's a very good bet. So like most kinases, it's going to act on serine or threonine, and it actually acts on many different things. But actually, we're not going to worry about what too many of these are right now, but many of these things are the sort of thing that you would potentially want to happen if you were suddenly startled and had to run away. It's going to be things that are going to give you energy. It's going to be things that are going to make the ability to make more epinephrine for you and things like that. So we're actually going to focus kind of on this one, phosphorylase B kinase. That is going to lead to the breakdown of glycogen. So the breakdown of glycogen is going to give you energy because it's going to give you sugars that you can break down for fuel. All right, we've seen this phosphorylase B kinase. It was the first kinase that we saw. So this is sort of an overview then of one portion of what this epinephrine pathway can do. At the pKa step, it's going to branch off and do a lot of other stuff, but this is just one, and we can see how this is eventually going to give us a very high concentration of blood glucose, which gives us the energy to be able to run away or fight, depending on whatever we're doing. So here we start with this is also showing us the amplification. We have one molecule of epinephrine. Now, usually you're actually going to have many more than that, but there might only be one that binds to a particular cell. And so whatever it is, you can just see that it's going to amplify by this much. So it binds the complex, and then the GS alpha subunit is going to go over and activate adenylyl cyclase, creating CAMP. Okay, but we can see even already here, this is going to happen multiple times, so maybe about 10 molecules. And then the CAMP can make about 200 molecules, so maybe like each one is making 20. But if there are 10 molecules going to that, that's going to be 200 total. And then that's going to activate more molecules of pKa. pKa is going to go activate this phosphorylase B kinase. So we have an even bigger number here. That's going to activate glycogen phosphorylase. This is actually the one that's going to cleave the glycogen, but this is going to activate the molecule that cleaves the glycogen. So glycogen phosphorylase A is actually the one that does the cleaving. That is going to act on glycogen to make, so it actually makes glucose 1-phosphate first, begins its cleave to glucose. And we can end up with this pathway because it's got so many steps, going from 1 molecule of epinephrine to 100,000 molecules of blood glucose. So these numbers are obviously somewhat idealized, right? They're not necessarily perfect, but it can just show you just how much we can amplify. So termination is going to happen, of course, when the signal stops. So again, if we had a situation like, a car sort of cut in front of you and you have a brief flash of this, but then you realize, okay, everything's fine, I'm fine, right? Your body is going to very quickly stop producing the epinephrine, and then any pathway that started is going to turn off. And this is relatively simple. It's just sort of each step will kind of, as soon as it's not being activated, it will stop happening. So eventually this will fall off as the concentration of epinephrine gets lower out here in the liquid. It will fall off any that it's already bound to. These G sub alphas, GS alphas will end up moving back and not getting another GTP. Then the adenylyl cyclase will obviously stop working. CAMP got created, right? It's going to activate PK and whatnot. So, but after a while, this actually gets cleaved back to AMP. So there is an enzyme that is going to, this actually starts happening as soon as the CAMP is around, but as long as you keep making more, it doesn't happen to a very big extent. But CAMP is going to get degraded. It basically just cleaves that bond. So here we can sort of see, right, all we're just going to do is just going to cleave this bond right here, and then we're left with just regular AMP instead of CAMP. So as the CAMP level lowers, then the pKa level is going to lower, and all the other steps will slowly stop too. However, again, desensitization can happen while the stimulus is still there. So even if you still have epinephrine out there, let's say you live in a place where there's just tigers around all the time, and I mean that would suck, but also you can't just be constantly terrified. It just is not a way to live. And your body, like the epinephrine response is just not a healthy way to be for a long period of time. So the benefit of being on high alert for the tiger would be sort of problematic in other ways because your heart would be beating really fast and things like that. So your body also is going to eventually shut it down. This also might be, let's say your body inappropriately makes epinephrine at times it really shouldn't, right? Or it makes way more than it should or something. You can't go through life just scared all the time and your heart beating really fast, so it's going to have to desensitize it. So the way this happens is basically it's going to pull the epinephrine receptor into the cell so it can't react. So if you put it in here into a little vesicle, it's not out here to bind to anything or to even act at all. So that's going to turn it off. So basically the way this happens is that this protein BARK, so B-A-R-K, gets recruited to here when that starts. And so essentially what happens is when the alpha section leaves, the rest of it says, hey, hold on, that guy left, so BARK should come over here. BARK, also a kinase. This is the beta-adrenergic receptor kinase, so it's going to put phosphates on the beta-adrenergic receptor. It puts these phosphates on the end here, and that is going to cause beta-arrestin to come, and eventually that is going to cause it to get endocytosed. So basically this entire section of the membrane is going to sort of join here. It's going to make a little vesicle that's going to literally put them inside the cell. Again, however, that you don't want that to happen forever. Otherwise, you would get one epinephrine response and never again, and that also would not be good. So then again, after a time, the beta-arrestin falls off, and another pathway can just bring this guy back out to the subsurface. So after a while, you're back to, you can get, scared again. So second messengers like CAMP are often used in multiple pathways. So this is just a list of some of the pathways that use CAMP as a second messenger. And this is one of the examples of that modularity. So CAMP can work on certain things, and any pathway where you might want pKa to get upregulated should just use maybe CAMP as a second messenger, instead of having, hundreds of different second messengers all for each different pathway. So another thing about these is I think we can tend to sort of think of everything as just sort of floating around in the cell, but they really need to be pretty near each other, especially for a pathway like the beta-adrenergic receptor, where it happens so fast, right? I mean, anybody who's had an epinephrine jolt knows just how fast this happens. I mean, this is super fast between this stuff getting created and the downstream effects happening. And so it kind of has to happen fast, because if it took 5 minutes for this to happen, the tiger has eaten you already. So way too late. Right, so it has to happen really fast. Other pathways maybe don't happen to happen as fast as this one, but many of them still do. So one way to do this is to hold them close to each other, so they don't have to go very far, and also so they can't kind of get lost on the way and distracted. So this ACAP here, we already saw part of this with the pKa, but ACAP is just a protein that stands for a kinase anchoring protein, and 5. So this is just going to anchor everything. So really, it's known for anchoring the kinase, but it also is usually actually attached also to the beta-adrenergic receptor and the adenylyl cyclase. So it's holding all of this stuff in a very close proximity so that everything can happen really, really fast, right? So as soon as the CAMP gets created, oh, look, pKa is right here, right? And then the target protein, for the pKa, that may not be quite as close. It's a little bit farther away, but everything's pretty close so that it can happen fast. All right, so I don't want to get you the impression that this is really the only one, right? So like I said, thousands of different, at least 1,000 different ones in the human body, more if we count other organisms. And so here's just one other example. We're not going to talk as much about this one, but this is a different GPCR pathway. And this is going to use a different set of second messengers that I just wanted to clarify here before we got to that. So this phosphatidylinositol 4,5-bisphosphate, this is just a lipid that we have seen before, right? It's a glycerophospholipid, and it's got this inositol head group on it. It's got a couple phosphates on it too. So what's going to happen here is we're going to cleave this right here, giving us diacylglycerol, which is basically a glycerolipid, but without any head group, even a phosphate, and then inositol 1,4,5-trisphosphate, right? Because we left the phosphate on there, so now it's got 3. In the lipid, it's sort of like this is considered part of the lipid, so that's why it's still bis, but it had 3. It's just where you consider it being part of. All right, so this guy is going to stay in the membrane, but this is now not a lipid anymore, and it is going to float off into the cytosol. All right, so in this case, and this is one that can be used in a couple different hormone receptors, so we're just going to kind of broadly call this a hormone is our signal. Same thing happens with the G protein, so all the dissociation and whatever. The effector enzyme here is obviously not going to be adenylyl cyclase, because that would make CAMP, but it's going to be phospholipase C, right? So phospholipase means it cleaves one of those phospholipid bonds, and it's going to cleave into now this diacylglycerol and the IP3. So IP3 is going to go over here. That's where that went. So this is the IP3 again. All right, so how this is going to work is so it's actually both of them are going to matter. IP3 is going to move over and bind to a calcium channel. This is a receptor-gated or a ligand-gated calcium channel. We saw a voltage-gated calcium channel, but this is a receptor-gated one, which means when IP3 binds, the channel opens up. And this is in the ER, the endoplasmic reticulum. And the Circa pump has been busily shoving calcium into the endoplasmic reticulum, so the concentration is much higher in there than out in the cytosol. So when we open up this channel, calcium flows out. All right, and so calcium is actually also called a second messenger. So there's really 3 second messengers here. We've got calcium, which is, we could hypothetically have called this a third messenger, but we just don't. Any small molecule that is created, not an enzyme or something like that that might be activated, but any molecule that's created later in a pathway is called a second messenger. So calcium is, so is IP3, and so is diacylglycerol. And what calcium does, calcium goes over here and activates a kinase called protein kinase C. So again, similar to protein kinase A, but different thing could act on different things. It's just a kinase. Diacylglycerol can also actually activate protein kinase C. And this is an example of that, how the pathways can intersect. These both, it's better if both are actually activating. And so then this protein kinase C just leads to phosphorylation of later proteins that is going to lead to whatever that cellular response is going to be. So in this case, we're not going to worry as much about what the downstream response is, but we can tell that it comes from the protein kinase C actions. All right, so that is GPCRs, and next time we will come and start in on the next sets of receptors.